Have you ever wished scientists could fix a typo in DNA the same way we fix a typo in a document? That's basically what base editing is. Traditional CRISPR gene editing works like molecular scissors. It cuts both strands of DNA and lets the cell repair the damage. But base editing is different. Instead of cutting the DNA completely, it just changes one letter of the genetic code directly. And that matters because many genetic diseases, like sickle cell disease, are caused by a single mistake, a single wrong letter. There are 4 DNA bases: A, adenine, T, thymine, C, cytosine, and G, guanine. Sometimes just one wrong letter can completely change how a protein functions. So base editing allows scientists to convert one base into another with incredible precision, erasing a spot instead of ripping out an entire page, essentially. So how does it work? Scientists use a complex using a guide RNA, a modified Cas9 protein that only cuts one strand of DNA instead of both, and a deaminase enzyme that chemically changes one base into another. For example, a cytosine, the base C, can be changed into a thymine. That tiny correction can restore the function of an important gene, and this technology recently achieved something historic. In 2025, doctors at the Children's Hospital of Philadelphia and Penn Medicine treated the first patient ever with a fully personalized CRISPR-based editing therapy. The patient was a baby named KJ, who was born with a rare and life-threatening genetic disorder called CPS1 deficiency. His body couldn't properly remove ammonia, causing toxic buildup that could damage his brain and liver. Now, normally patients with this disease require a liver transplant, but KJ was too young and his body was too unstable for that procedure. So, researchers created a therapy designed specifically for his exact Within just 6 months, scientists developed a custom base editor delivered through lipid nanoparticles, which function as a cellular delivery vehicle, directly to his liver cells. The editor corrected the faulty DNA letter responsible for the disease, and the results were remarkable. After treatment, KJ could tolerate more protein in his diet, needed less medication, and recovered better from illnesses that could have previously been deadly. Doctors reported no serious side effects from the therapy. Now, this wasn't just a breakthrough for him. It showed something much bigger. Personalized gene editing could completely change how we treat rare diseases. Genetic disorders like KJ's are so uncommon that pharmaceutical companies often don't develop specific treatments for them. Creating a new drug can take years and cost billions of dollars, and for ultra-rare diseases that only affect a small number of patients, companies don't see enough financial incentive to invest in a treatment. Personalized base editing can change that model entirely. Instead of creating one massive drug for millions of people, scientists may be able to design highly targeted therapies for individual mutations, even for diseases so rare that only a handful of patients in the world have them. KJ's treatment showed that researchers can potentially build a custom therapy in months, tailoring the edit to the patient's exact DNA error. For families affected by rare genetic diseases, that possibility is life-changing.